Recombinant DNA is DNA created artificially by combining the DNA from two or more organisms into a single recombinant molecule. DNA recombinant technology is the transfer of a segment of DNA from one organism to another organism where the DNA reproduces. Recombinant DNA technology involves several steps in a specific sequence. The steps are isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at a large scale, and extraction of the desired product. Recombinant DNA technology begins with the isolation of a gene of interest, that is the donor DNA. This can be achieved by cutting the DNA with the enzymes. To cut the DNA with the restriction enzymes, the DNA needs to be in pure form, free from other macromolecules. In eukaryotes, DNA is enclosed within the membranes. So we have to break the membranes to release DNA along with other molecules like RNA, proteins, carbohydrates, and lipids. This can be achieved by treating the bacterial or plant or animal cell with enzymes such as lysozyme for bacteria, cellulase for plant cells, and chitinase for the fungus. RNA can be removed by treating with ribonuclease. Proteins can be removed by treating with protease enzymes. Other molecules are removed by suitable treatments, and purified DNA precipitates out after addition of chilled ethanol. In the second step, cutting of the DNA at specific locations is to be done. And to do this, a non-DNA sequence from the donor cell is identified and removed with a restriction enzyme. When cut, each fragment of DNA has an overhanging piece of single-stranded DNA on its ends called the sticky ends. With the help of these ends, the fragment is able to attach with any DNA molecule that contains a complementary sticky ends in the presence of enzyme ligase. The fragments of DNA can be separated using agarose gel electrophoresis. Because of its phosphate groups, DNA is negatively charged, and when it is placed in a semi-solid gel and an electric field is applied, the DNA molecules migrate towards the positive pole. Polymerase chain reaction is used to produce identical copies of a short DNA sequence. This process includes 3 stages. A DNA molecule of interest is denatured by heat at 90 to 95 degrees Celsius. The 2 strands separate due to breakage of the hydrogen bonds holding them together. In the presence of an excess of nucleotide, oligonucleotide primers are added. The primers are complementary to either end of the target sequence but lie on opposite strand. As the mixture cools at lower temperature, that is 50 to 60 degrees Celsius, each sequence of DNA molecule becomes annealed with an oligonucleotide primer. DNA polymerase, isolated from a bacterium Thermus aquaticus, is then added and complementary strands are synthesized. The polymerase causes synthesis of new strand in the 5' to 3' direction. If this process is repeated many times, the segment of DNA can be amplified to approximately billion times. That is 1 billion copies are made. The amplified fragments, if desired, can now be used to ligate with a vector for further cloning. The recombinant DNA is introduced into the host cell in a process called transformation. The recipient cells, after making them competent to receive, take up DNA present in its surrounding. If a recombinant DNA possessing antibiotic resistance gene is transferred into E. coli cells, the host cells become transformed into the antibiotic resistant cells. If we put transformed cells on agar plates containing ampicillin, only transformants will grow, untransformed recipient cells will die. The ampicillin resistant gene in this case is called the selectable marker. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial or a plant or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. So there is a need for the recombinant DNA to be expressed. The foreign gene gets expressed under appropriate conditions. The expression of foreign genes in host cells involve understanding many technical details. After having cloned the gene of interest and having optimized the conditions to induce the expression of the target protein, one has to consider producing it on a large scale. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiologically most active phase. This type of culturing method produces a larger biomass, leading to higher yields of desired protein. Small volumes of cultures cannot yield appreciable quantities of products. So, to produce in large quantities, the development of bioreactors where larger volumes like 100 to 1000 liters of culture can be processed. So this kind of bioreactors were required. Thus, bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, using microbial, plant or animal or human cells. A bioreactor provides the optimal conditions for achieving the desired product by providing optimal growth conditions like temperature, pH, substrate, salt, oxygen or vitamins. The most commonly used bioreactors are of stirring type. A stirred tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. The bioreactor has an agitator system, an oxygen delivery system, and a form control system, a temperature control system, pH control system, and sampling pot so that small volumes of the culture can be withdrawn periodically. This is the figure of a spranked stirred tank bioreactor through which sterile air bubbles are sparged. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The processes include separation and purification, which are called as downstream processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo thorough clinical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product.